Okay, so now that we've identified all the organelles that we'll find in a eukaryotic photoautotrophic cell, let's take a look at each one in a little bit more detail. And in this series of 6 slides, we'll be looking at the nucleus and ribosomes and this system known as the endomembrane system, which includes the nuclear envelope, the endoplasmic reticulum, Golgi apparatus, and we'll also be taking a look at vesicles and vacuoles and some specialized vesicles and vacuoles and what's the difference between vesicles and vacuoles. All right, so here we're starting with the nucleus, and we're just going to go ahead and label the components of the nucleus here just to remind you. And it's a good exercise for you to do this too. It'll help you remember the different components. So we've got the nucleolus, which, if you'll recall, is the site of ribosome synthesis, or where ribosomes are made. Surrounding that, we have the chromatin, which, if you'll recall, is a combination of DNA and protein. The proteins help to organize the DNA and control its expression. Surrounding the whole nucleus, we've got the nuclear envelope, and it's an envelope because there are 2 phospholipid bilayers that make it up. If you look closely at the diagram, the outer one's a little darker blue and the inner one's a little lighter blue, so they're actually showing you that there are 2 layers to the nuclear envelope. It's a double membrane. If the nuclear envelope looks like Swiss cheese to you, that's because it has holes in it, and in biology we call holes pores. So because these pores are in the nuclear envelope, we refer to them as nuclear pores, and they function along with the nuclear envelope as a selective barrier, and so certain things are going to be able to pass through those pores while other things can't. Most notably, DNA cannot pass through the pores, or does not pass through the pores, whereas RNA does. So RNA is able to leave the nucleus once it is made through the process of transcription in the nucleus. It's able to leave and go out into the cytoplasm. Remember that this nucleus is surrounded by cytoplasm. And also on the nucleus, and you'll need to draw these in because they're not already on your diagram, I just wanted to remind you, like it was pointed out on the previous diagram of the whole cell, there are bound ribosomes on the surface of the nuclear envelope. And those are to make proteins, most specifically proteins that are going to enter the nucleus and operate within the nucleus. So hopefully it's clear that the most important compound contained in the nucleus is deoxyribonucleic acid, or DNA. And as I pointed out, it doesn't leave the nucleus. It stays in the nucleus. The following describes gene expression. If you'll recall, that's what we call everything from gene to protein, the whole process of going from the information of a gene to making a complete protein. That's known as gene expression. And also it goes even further than that. The function of the protein, the outward phenotypic expression of that protein, or ultimately of the gene. That's really all of gene expression. And also the central dogma of biology. We refer to it as that because it is so fundamental to life. All life functions in this way, from genes to proteins. So the DNA nucleotide sequence spells out the genetic information of proteins. Sequence. I'm trying to emphasize that because it's all about the sequence. So nucleotide sequence spells out the sequence of amino acids in a protein. But in between the DNA sequence and the protein, amino acid protein sequence, there's RNA, right? Ribonucleic acid. So even though the DNA stays in the nucleus, the information of the DNA leaves the nucleus after being transcribed to RNA. And it's the sequence of nucleotides in RNA. And that's why we call this transcribing. We're transcribing the nucleotide sequence of DNA to the nucleotide sequence of RNA. So the type of, or the language, I guess you could say, is not changing. We're just transcribing it, copying it over from nucleotide sequence to nucleotide sequence. RNA then carries out the process of translation in the cytoplasm at ribosomes, and that's where the RNA nucleotide sequence is translated into the amino acid sequence of a polypeptide or protein. So we go from DNA nucleotide sequence to RNA nucleotide sequence to amino acid sequence in a polypeptide or protein. That's the central dogma of biology. That's how all life operates at the fundamental level. DNA is also the stuff of heredity. It's hereditary information that can be passed from cell to cell or passed from parents to offspring during reproduction. And there are two processes that are involved there, and they both refer to a different kind of nuclear division. Not cell division completely, but nuclear division. There's the process of mitosis, which produces two identical nuclei before cells divide, or before unicellular organisms reproduce by binary fission. Eukaryotic unicellular organisms anyway, because if they're prokaryotes, don't have a nucleus, so you can't divide what they don't have. Meiosis is the process, or meiosis is the process of nuclear division that results in sex cells, gametes, sperm and egg, which have half the number of chromosomes, and they're genetically unique. And we'll be studying both those processes in upcoming units. But the bottom line for the nucleus is that it functions in gene expression and heredity. So that's where you want to begin, but you need to be able to then explain the details of what that means, gene expression and heredity, which are basically the functions of DNA. So here we have the ribosome, and ribosomes are made up of both ribosomal RNA and 80 different proteins. So there are genes for these functional RNAs that partially make up the ribosome. And if you'll recall, there are two different subunits to the ribosome, large subunit, small subunit. And the ribosomes basically act as a platform for the other RNAs to come together and string amino acids together to make a polypeptide or protein. So here's the messenger RNA, and the nucleotide sequence of the messenger RNA. Every 3 nucleotides is a codon. The codon is being matched up to anticodons on transfer RNA, if you'll recall, because we already studied this. And each transfer RNA is bringing along an amino acid that's being strung together into this polypeptide that is growing and coming out of the top of the ribosome in this orientation anyway. So that, in a nutshell, is the function of the ribosome that you should already be aware of. Again, ribosomes are free in the cytoplasm or bound to the nuclear envelope or the rough endoplasmic reticulum, which we'll be looking at in more detail coming up. And they function as tiny protein factories, basically, by the process of translation. So this process that I just described is protein translation. And it's called translation because we're translating the nucleotide sequence of RNA, messenger RNA here, to the amino acid sequence in the polypeptide or the protein. And so basically we're changing languages. We're translating from the language of nucleotide sequence to the language of amino acid sequence, and that's why it's known as translation. So during translation, the nucleotide sequence of a gene that was transcribed to the nucleotide sequence of messenger RNA is translated into the amino acid sequence of a polypeptide or protein. And what that little statement describes is what we refer to as the central dogma of biology, because it is so fundamental to life. So the bottom line, ribosomes function as the site of protein synthesis. Synthesis just means making, so that's where proteins are made. But that process of protein synthesis is known as translation. And that brings us to the endoplasmic reticulum, which is quite a mouthful, so we often just call it ER. But endoplasmic reticulum comes in two forms. There's rough endoplasmic reticulum and smooth endoplasmic reticulum. And the difference is whether or not it has ribosomes. Rough endoplasmic reticulum looks rough under the microscope because it is covered with ribosomes, bound ribosomes. The smooth endoplasmic reticulum does not have bound ribosomes. So in this diagram, we're again looking at rough endoplasmic reticulum and actual colorized electron micrographs showing the ribosomes. So both ERs are responsible for synthesis, but they're responsible for the synthesis of different organic biological compounds. When it comes to the rough endoplasmic reticulum, it handles primarily proteins, while smooth endoplasmic reticulum handles primarily lipids. So, for example, because the rough endoplasmic reticulum has ribosomes, protein would be made at a ribosome, and that protein would go inside the endoplasmic reticulum, and inside will be an environment and enzymes that will act on that protein that has just been made, to cause it to change in its conformation. Remember, that's what we call the shape of the protein. So the protein might be modified. It might be cut shorter. The chain of amino acids might be cut shorter, or it might be joined to other polypeptides to make a complete protein, because remember that some proteins are made up of more than one polypeptide. So it can be modified in a variety of ways before it is a functional protein for the cell. And that takes place within rough endoplasmic reticulum. Smooth endoplasmic reticulum has the enzymes necessary for producing the variety of lipids that the cell needs in order to build its structure, because a lot of the structures within a cell are membrane bound, right? There's membrane everywhere, from the plasma membrane through, the structures of the endoplasmic reticulum and the nuclear envelope. It's all membrane. So once these proteins or lipids are ready to go, they're typically packaged into transport vesicles and shipped out to the Golgi, which is the next organelle that we'll take a look at. But the nuclear envelope and the endoplasmic reticulums and the Golgi are all part of what is referred to as the endomembrane system. So here we see that, and you have to draw this in because it's not part of your diagram. This is representing the production of a transport vesicle from the ER. So once the products of the endoplasmic reticulum are ready to be shipped out, they're packaged into little membrane-bound vesicles that contain those proteins and or lipids, and they can contain other things too, even carbohydrates, to be shipped to the to be shipped to the Golgi apparatus. So we'll refer to this as an ER transport vesicle, because when we look at the Golgi, we'll also be looking at the fact that Golgi produce Golgi transport vesicles. So, like I pointed out, the nuclear envelope, rough and smooth endoplasmic reticulum are continuous. Notice the connection here between the nuclear envelope and the rough endoplasmic reticulum, and then the rough endoplasmic reticulum is kind of transforming into smooth endoplasmic reticulum, and it's all connected together, so continuous, and functions as part of the endomembrane system of the cell. So the bottom line is protein or lipid synthesis, depending on which ER you're talking about, processing, in other words, changing those proteins and lipids in some way within the endoplasmic reticulum in that environment inside the membrane sacs that make up the endoplasmic reticulum, and then transporting it in transport vesicles, ER transport vesicles, typically to the Golgi apparatus, but it might be ready to go someplace else in the cell. So not all ER transport vesicles are bound for the Golgi apparatus. So the Golgi is often just referred to as the Golgi, but it's also often referred to, as I just mentioned, the Golgi apparatus. It can be referred to as the Golgi bodies, because each of these membrane sacs that we see here that are stacked up to produce the Golgi, and by the way, this diagram is cut open, so the membrane sacs are cut open so that you can see that they have a cavity on the inside, or what we call a lumen on the inside, a space on the inside, where, again, enzymes are going to act on what is being processed by the Golgi. But it's described as a stack of membrane sacs. The membrane sacs are known as cisternae. So each one of these is a cisternae, and it functions as part of the endomembrane system, as I mentioned, because it receives proteins and lipids and other compounds from the endoplasmic reticulum. Those compounds are then going to be modified and sorted and packaged and shipped from the Golgi, and that's what these vesicles are all about, in Golgi transport vesicles. So if we carry on from the previous slide, that's what this arrow is representing. We've got an ER transport vesicle that's coming from the endoplasmic reticulum, and it is going to merge with the cisternae, the first cisternae in the Golgi apparatus. And that's where these cisternae come from. They come from the merger of transport vesicles that are coming from the endoplasmic reticulum. The transport vesicles merge together and produce these sacs of membrane that have a lumen inside. So they merge at what's known as the cis face. This end of the Golgi is a cis face, and don't worry, we're going to label it in a minute. But that's what forms the cisternae. The cisternae then, they're formed at this end of the Golgi, and as new ones are formed, the older ones move through the Golgi from the cis to what's known as the trans face of the Golgi, trans, where they finally bleb into transport vesicles. And that's actually a term that's used a lot in biology, just to refer to what's going on here, where a membrane kind of pinches off and forms vesicles or small, round, lipid, membrane-bound lipid compartments. And they're going to contain the final product, final protein, final lipid, final carbohydrates, whatever. So just to run through it again, we'll label everything here. So each one of these sacs is a cisternae, and they form at the cis face, which makes sense. Cisternae, cis face, that's where the cisternae form. They mature as new cisternae are formed at the cis face. The older ones are moving through the Golgi apparatus towards the trans face. And the trans face makes sense too, because transport vesicles are blebbing or being created from the trans face. And they can be secretory vesicles, which means that they're bound for the plasma membrane to be secreted outside the cell by exocytosis, if you'll recall. And so the final proteins and lipids are transported where they're needed in the cell or to the plasma membrane for secretion through exocytosis outside the cell. So the bottom line for the Golgi apparatus: protein and lipid modification, sorting, packaging, and shipping. That's what the Golgi apparatus does. If you look at this diagram, notice that we've got the Golgi apparatus here, and it's producing vesicles, and these vesicles are specialized vesicles. So we're going to take a look at them because they're different than other vesicles produced by the Golgi, because they contain digestive enzymes and acids. So you can kind of think of these vesicles known as lysosomes, and they're called lysosomes. Some means body, lys means to burst, or break. And in this case, they're breaking down compounds that are contained within other vesicles. So they have hydrolytic enzymes. Remember, hydrolysis is the process of breaking polymers apart, breaking the bonds between the molecules that are making up polymers. And they break down all of the 4 classes of organic biological compounds. So they can break down lipids, carbohydrates, nucleic acids, and proteins. Those are the 4 classes of organic biological compounds, and their lysosomes have hydrolytic enzymes that are able to break down all of those. You can also think of these as digestive enzymes. Hydrolytic enzymes, digestive enzymes, they break things down. So here in this scenario, we're seeing a food item being brought in by the process of phagocytosis. If you'll recall, that's a form of endocytosis, and phage means to feed. So phagocytosis is a process of cell eating. So it's bringing in a particle that is way too big to come through the phospholipid bilayer of the plasma membrane. So it has to be done through bulk transport, phagocytosis, where the membrane, the plasma membrane, kind of pinches in and engulfs the food particle and surrounds it in membrane, and that forms a food vacuole. Notice that the lysosome being produced from the Golgi is going to fuse with the food vacuole, and that produces what's known as the lysosome, when it's first made from the Golgi, is known as a primary lysosome. So this is a primary lysosome. But it's going to fuse with the food vacuole, and when that happens, it empties its digestive enzymes and acids into the food vacuole and starts to digest the food. So it starts to break down the proteins and carbohydrates and lipids and nucleic acids that make up that food item. And when that happens, it's known as a secondary lysosome. So once the primary lysosome fuses with the food vacuole, that one structure that results from that is known as a secondary lysosome, and that's the structure in which digestion is taking place. And the nutrients then are going to diffuse out of the lysosome into the cytoplasm to be picked up and used by the cell. So in that way, cells can gain nutrients. So that's this side of the diagram, but you'll notice that there's something else going on this side of the diagram, and if you look closely here, you might recognize this as an organelle, more specifically mitochondria. So organelles wear out and they break down. They stop working properly, and so they need to be broken down. They need to be digested, and that's a process known as autophagy. Auto means self, and again, phage means to feed. So this is feeding on self, breaking down components of the cell, breaking down organelles that are no longer functioning properly. So the components are engulfed by a primary lysosome, and that produces a structure known as a phagosome. So the only difference between a secondary lysosome and an autophagosome is what is being digested. In a secondary lysosome, something is being digested that has been brought into the cell from outside. In an autophagosome, a component of the cell itself is being broken down because it has worn out, and it's going to be recycled. The nutrients are going to diffuse out of that autophagosome and be used by the cell. One other function of these lysosomes is that sometimes cells are supposed to die. It's known as programmed cell death. This happens a lot during development. For example, at one point during your development, you have webbed fingers and toes. You have structures that develop that then eventually go away as you develop. And that's done by the cells that make up the webbing between your fingers, for example, dying. They're programmed to die at a certain time during development and go away. This process is also known as apoptosis or apoptosis. Some people go ahead and pronounce that P. I prefer to leave it silent. So apoptosis, programmed cell death. That's when lysosomes break open inside the cell. So when a cell gets a signal to kill itself, basically, to commit suicide, you could say, it breaks open its lysosomes inside the cell, and that digests the cell from the inside out and kills the cell. So the bottom line, there are a lot of different functions going on here, basically 3 different functions. We've got intracellular digestion of food, so to speak. We've got intracellular digestion of organelles, worn out organelles, and we've got this process of cell suicide or cell death. So the bottom line is intracellular digestion, autophagy, and suicide are the functions of lysosomes. So you may be wondering, we've been talking about vesicles and vacuoles and throwing around those words a lot, so we're going to here look at the difference between them, general difference between them, and generally vesicles are smaller and they transport things. So they're small transport compartments, whereas vacuoles are large storage compartments. That storage may be temporary, but they still are functioning in storage at least for a period of time. So in plant cells, if you'll recall from that diagram, this is the same diagram that we labeled on the earlier slide. The central vacuole is this huge vacuole that sits in the middle of the cell. That's why it's referred to as central. And it stores water and minerals. And it supports the cell. Its secondary function is to support the cell because the water that is in there is under pressure. The pressure produced by osmosis of water into the central vacuole. The solution inside the central vacuole is hypertonic, if you'll recall that term. It's hypertonic compared to the solution outside. Therefore, water will osmose into the central vacuole and produce pressure. Just like blowing up a balloon. It produces pressure inside the central vacuole, and that pressure is known as turgor pressure. It's such an important concept in plant biology that it's been given its own name, turgor pressure. Turgor pressure is produced when the plant is in its preferred hypotonic environment. So in other words, when you water a plant, you're surrounding the plant with fresh water. The cells are surrounded with fresh water, and that fresh water is hypotonic compared to the hypertonic solution inside the cell. And water will diffuse by osmosis from hypotonic to hypertonic. So wherever the hypertonic solution is, water is going to diffuse to that solution, towards that solution. And this would be a solution that has a lot of things dissolved in it. So the central vacuole, in being a storage compartment for not only water but also dissolved minerals and things, that means there are a lot of dissolved solutes in that solution. That's what makes it more hypertonic than the solution outside, and water therefore osmoses into that central vacuole. So again, it produces osmotic pressure, which has the special name known as turgor. That helps support the cell, and the plant is made up of hundreds of thousands to millions of cells, and so it helps support the whole plant. We call this state turgid after turgor pressure. So it's in a turgid state when the plant is not wilted. It's being well supported by the pressure within the central vacuole. We call that plant in a turgid state. If for some reason, for example, you don't water a plant, then it no longer has enough water and pressure within the central vacuole, and the plant will wilt. But we call wilting, more technical term, flaccid. So the cell becomes flaccid, and the whole plant becomes flaccid. So that's the central vacuole of plant cells. This is a protozoan. You may recognize it as a paramecium, and we're going to be studying protozoa in this unit. And protozoa, many freshwater protozoa, because they're surrounded by fresh water, and that term fresh water is just like the opposite of salt water. Fresh water is hypotonic, compared to the cytoplasm inside the cell. Therefore, water is going to diffuse by osmosis into the cell. So that means protozoans that live in fresh water are constantly under threat of pressure building up inside the cell. And unlike plant cells that have a cell wall, protozoan cells don't have a cell wall, therefore they can't stand high pressure. They can't stand that buildup of pressure. It could eventually cause the cell to explode, to pop, and therefore die. So they have these things called contractile vacuoles. And contractile vacuoles are an example of very temporary storage. They take on water, they take in water, so they're like these channels that are going out from the contractile vacuoles. And notice that there are two of them in this diagram. And the channels are basically collecting water, helping to collect water. And when you look at these under the microscope, you can actually see them expanding as they take on more and more water. But then eventually you'll see them contract, squeeze. They pump the water out of the cell. There's a pore through which the water is going to go from the contractile vacuole out across the plasma membrane and out of the cell. So you can think of these as kind of like sump pumps. If you have a sump pump in your basement, when water comes in, when it rains, the sump pump pumps it back out of your basement. This is doing the same thing. When water is osmosing into the cell, these contractile vacuoles collect that water and contract and pump the water out of the cell. And in that way, it's carrying out osmoregulation, the regulation of water balance in the cell. And your body does the same thing, but it does it with your kidneys, which are part of your excretory system. But so this is like one of the most primitive examples and most obvious examples of water regulation, of osmoregulation within a living thing in what is arguably considered a very simple organism, protozoans. So when it comes to vacuoles, there are three kinds of specialized vacuoles that you're responsible for knowing. One being the central vacuole of plant cells, another being the contractile vacuole of freshwater protozoa, and then a third being the food vacuole that we saw in the previous slide. That's a vacuole. It's, again, it's more for temporary storage, kind of like the contractile vacuole is temporary storage of water. The food vacuole is temporary storage of food until it can be digested in a secondary lysosome. So the bottom line, for both vesicles and vacuoles, we're talking about storage, support, water balance. Oh, I'm sorry, not for both, just for vacuoles. Just for vacuoles is storage, support, and water balance. I'm sorry.